Well, good morning, everybody. Thank you very much. I'd like to thank CRB and for the invitation to come talk to you today about some of our progress. This presentation is more like a summary of the different projects that we've been working on, a number of projects which directly have direct application and directly impact to the citrus industry every season. So there's a number of pre- and post-harvest diseases of concern in our worldwide marketing of our crop. We have Septoria Spot, many of you are familiar with our trade with Korea. We have Phytophthora brown rot caused by multiple species that are quarantined species, just like Septoria, with other trade partners like China. And we also have some new diseases that are coming into the state. We can call them invasive. They've been known in other parts of the country, but the sweet orange scab has been detected in California. And this, I'll mention a few words about this. This is another quarantine pest in some trade markets like Australia, New Zealand, and Vietnam. We continue to look at post-harvest decay management. Obviously, we see fluctuations in the penicillium species that are causing decay. And we've seen Geotrichum citrii, the citrioranthae, the sour rot pathogen also increasing at certain times of the year. So part of our research is to improve the quality and standards. And what we were listening to Casey earlier this morning, and I think this is what separates us from the rest of the citrus world market, is that we can provide the highest quality fruit to our export markets. So for Septoria Spot, many of you know I've been running the NAVIC program. This disease we've started way back in the mid-2005, and we've worked on it. We've shown that the pathogen lives in trees and leaves, fruit, and twigs. And when it gets onto the fruit, a healthy fruit is immune to the disease. And so what we've identified is that it's cold injury or frost damage or cold injury that causes little marks on the fruit, and they can be small, but that's the site of infection by the Septoria pathogen. Now, one of the big things that we also identified, and I was part of the trade agreements. Fortunately, the citrus industry sent me over to do the negotiations with the Koreans, and working with the APHIS personnel, we were able to negotiate the latest version of the working plan that we have with the Koreans, that they have to detect the actual fruiting bodies and sporulation of the fungus, which is shown in the upper right. And so we can detect it using molecular methods. They have to use actual symptoms for the detection of the disease, and that's given us a really clear advantage. And we started working on anti-sporulation fungicides, because if you put a fungicide on that prevents sporulation, even if you have some fruit that go through that have an ice mark on it, the Koreans won't see sporulation, so it can't be rejected. So that's one of the big things that we contributed on, and we've been working on the NAVIC program for a number of years. Many of you know we provide forecasts for the disease. We use this model system on the right-hand side, like that chart. We accumulate precipitation, and we look at the number of hours that the fruit is exposed to -1 centigrade. And we can do that for all the different cinder stations. And we've started with Fresno and Tulare counties, and then we expanded, and now we're covering pretty much all counties, citrus counties in the state. So we can make a forecast, and then we can distribute that to the industry with advisories for the second sprays. Basically, the NAVIC program is helping to identify lots that are free of disease, and if we do detect it, then we can divert it to domestic markets or other markets that don't have this quarantine in place. So we didn't stop there. One of the big things is, as I point on the left-hand side, we currently are doing this program. We monitor for the weather, we make forecasts, we have samples come in. we tell people that remind people that between October 15th and November 30th is the time when we want to put our first prophylactic protective treatment on. And then if the weather is wet and cold, we can put out another advisory when to spray the second spray in January and February. Last year was very warm and dry. We delayed that all the way to the end of February, so it saved sprays. People weren't actually applying fungicides because there was no need to. And subsequently they realized there's no pressure, so they didn't send samples to the Maverick lab either, but it's a good thing for the industry because they, again, saved money. But what we're doing on the right-hand side is that we're looking at this flow chart, and there was two concerns that the industry had. They told us that they've been spraying routinely for many years, so what we wanted to do was to verify that we could reward people if they put on a spray in the mandated spray in October, November, and then they send a Maverick sample and it's negative after January 1st, then they don't have to put a second spray on, they're exempt. So this is rewarding for good management practices. The second thing we wanted to do here from the industry was they were concerned that if a lot was positive, it eliminated it from the chances to go in Maverick, to go to the Korea program. So what we did is we said we have all these, we worked for many years to get the registrations of anti-sporulation fungicides. So even if your lot is positive after January 1st, if you put the second spray on with an anti-sporulation fungicide, it's still eligible to go to Korea. And so that's some of the exciting developments. So this increases the amount of lots that can potentially be traded, and this is ways to improve the NAVIC program. So with these different modifications to NAVIC, we can see that we could reduce the number of fungicide applications by rewarding people that have managed the disease, and we can keep Septoria Septoria lots that were positive for the disease in the program. And so those are some of the new aspects of the NAVIC program. This is currently going to be published in Citrograph, so people have a really clear understanding of how they can use this information to their benefit. So sending samples to the NAVIC lab is still profitable, because if you can save a spray, you can save literally millions of dollars for the industry. We continue to look at new fungicides. We're excited. These fungicides like Quadris Top has been around for a few years, but now we have Preaxor, Lunisensation registered, and you can see the high efficacy of these treatments. So we do have alternatives to copper. And again, alternatives to copper keeps phytotoxicity down, and it keeps environmental contamination to a limited amount. So we still wanted to practice common sense management practices with cultural practices to remove dead branches and limbs. But with labor costs, the most effective way is to use these fungicide programs effectively. And we have the first application with copper, and then we have the second application with one of these choice materials that we've identified, listed. And we're continuing to work with new ones. We have pending several new fungicides, Sepia, Diabla, and Riverside Duo. And then lastly, the post-harvest fungicide Graduate A+ and TBZ are both highly effective as anti-sporulation fungicides. The next disease I want to talk to you about is the Phytophthora diseases. And again, this is for our trade with China, and the pathogens of concern are the two bottom ones there, Phytophthora syringae and Phytophthora hibernalis. These are quarantine pathogens, and they're mixed in with some of the other pathogens that we see in citrus, Phytophthora nicotiana or parasitica, and Phytophthora citrophthora. So we decided to make the programs that we have for Phytophthora for all the Phytophthora diseases, because we didn't really want to focus on specifically identifying which one. We're just considering them all to be negative for potential quarantine issues. So Phytophthora can cause root rots, it can cause trunk mimosis, trunk infections, and it can also cause brown rot, an infection of the fruit. And this is of the most concern for the export market. Phytophthora has lots of ways to survive. This is a disease cycle. You can see that there has different types of spores. The idea here is just to illustrate that there's many different ways the fungus can survive. And it lives in the soil. It can cause root infections. It has long-term survival structures like oospores. But it can quickly multiply. One oospore can produce a sporangine, which could produce 30 or 40 zoospores. So it can go exponential in a disease curve very quickly. So we wanted to integrate, obviously, water management practices, but we can't control the rain. So our main focus, again, we turned to fungicides as a practical way to manage this. One of the things that we also did for the Chinese work plan was to develop models, and we do put co-advisories out for the industry on Phytophthora. We use various models, and here is illustrated two. One is a copper degradation curve, and we've generated data with the amount of rainfall, and we have a standard curve with how much reduction in copper occurs. And as we get to 50, 60, 70% reduction in copper due to rainfall, you can see that we get higher and higher risk, and that's those higher numbers in red. So the other thing we did was to follow the Septoria model. In the lower right, we have a model for forecasting Phytophthora syringae. And right away you can see that it's really the cool temperature between 1 and 10 degrees that is ideal for Phytophthora syringae, but it can go up to 20 degrees, and so the red has that little shelf to it, giving us the high risk. So when we accumulate, again, rainfall, and at these different temperature levels, we have higher risk for Phytophthora syringae. So we use those two to, again, make forecasts for the industry and put out advisories. When we do the Septoria advisory, we have a Phytophthora advisory as well. Not to stand still on anything, we were very fortunate to identify several new fungicides for Phytophthora that came onto the market in 2014 and 15 for vegetable crops. I immediately asked Syngenta and Valen USA to allow me to test these. We wrote a project for the CRB, and we were funded. And what the outcome is in 2018, 19, and 20, we were able to register Arandis, Residual, and Revis. And these are the first three new Phytophthora fungicides that were registered since Residual and phosphites in the 1980s. So this was a unique thing because each one of those has a different mode of action, and we can develop integrated management practices. One of the exciting things in those gray boxes is that for foliar application of Arandis, we're using 1 to 2 fluid ounces per acre. I don't know if you know what a fluid ounce is. It's about 20, 30 mils. And so we're talking about very small amounts of material spread over the entire acre, which is, again, environmentally sound. These are organic materials. They're based on carbon, and they degrade, and they're extremely effective against Phytophthora. Not to stop there, we also identified this Illumin, and we submitted that to the IR4 program in this past year, and it was accepted. So one of the experiments that we did is we were testing different materials, and we ran across phosphite resistance, and we know that their phosphites are really economical phosphonate fungicides, and people are using them extensively since about the late 1980s in the citrus industry to control Phytophthora. But now we're detecting resistance to Phytophthora and in Phytophthora populations. And so I'll say more about that momentarily, but what we wanted to do is test these new fungicides to make sure they work on phosphite-resistant isolates. And so we sprayed trees, and we did a tree with Revis, a tree with Arandis, a tree with copper. And then what we did is we harvest those fruit and we inoculate them. So it's like the most severe example over different time periods. And you can see some of the results. The check all comes down when we do this practice. But we can harvest that 1 week, 2 weeks, a month, 2 months afterward, and determine the performance of that fungicide. Well, lo and behold, we were at another citrus meeting about 2 years ago before COVID, when we had a previous in-person meeting, And we applied these materials, and you can see right away that the Arandis treatments at the different rates that we applied. Arandis Ultra is a mixture of Arandis with Revis, so that's going to be registered also. And then we had Revis, and even after 4 weeks, you know, we had 95% in the control, we have 1% in the Arandis Ultra. And after 8 weeks, it's only 2% infection. So these things have long residual activity, and we did this at the standard 400-gallon rate. We also did it at 100-gallon rates. Back to that meeting that we had, many applicators were asking us, can we do this with lower volumes and get the same results? Because we can go 4 times farther if we spray lower volumes and get the same results. And so sure enough, we've been doing studies the last couple of years to show that even when you're applying these fungicides, Arandis and Revis as foliar treatments, you can do it even at lower gallonage, and this is better for the applicator because he can go 2, 3 times, 4 times as far on a single 500-gallon tank load. Now you might want to ask, well, what about the MRLs? And so you see that the MRLs for Arandis, the oxythiopipeline, and the mandipropamide are very low. They're at 0.1, some are at 0.05, some are even at 0.01 in the EU, for example. The US is also low for Arandis at 0.05, but it's 0.5 for mandipropamide. And you say, well, how are we going to meet our export? So what we did is we did studies where we showed we applied Arandis at 1.2 and 2.4, which are labeled rates, and we were able to detect at harvest 0.01, which is right at the threshold of detection. But if we ran that fruit through the pack line, those are, again, 2 studies, one at UCR and one at Kearney. If we ran the study through those treated fruit through a pack line where there's brushing, sanitation, and other fungicide applications, we have non-detects. And so because we're using these extremely low rates, we're able to harvest that fruit, pack it, and we're ensuring the industry that there's not going to be any MRL violations. We did the same study with Revis, and similar results came out. Now, the phosphite resistance is an issue. What these graphs are showing is that phosphite resistance is shifting. So the taller bars on the left up there for these 2 Phytophthora species show there's a long tail to the right, and the red bars are showing resistance. And so what we're afraid of, if people keep moving, just using phosphites without rotating, this is part of an IPM program, just like they do with insects. If we rotate between different modes of action, we'll be able to prevent the selection for resistance. So again, when we're looking at the right, the B graph there, you can see that a large percentage of the population is shifted already to resistant levels. And phosphites used to work at 5 and 10 parts per million. And now we got isolates that are up to 300 parts per million before they're inhibited at an EC50 or effective concentration of 50. So we still want to keep phosphites. So the way to do this is to rotate between the different modes of action, which is a standard practice in disease and pest management. The bottom graph just illustrates that if we take a phosphite-resistant isolate, the red bars, and we try to control that with a pre-harvest treatment, and then harvest that fruit and inoculate it with a resistant isolate, the red bar is the same as the control. It means that this is field resistance. It won't work anymore. But if we use a different fungicide with a different mode of action, in this case oxythiopipeline, we get 100% or nearly 99% control. So with phosphite, we're looking at also ways to prevent this from getting to using it post-harvest. The Chinese don't want us to use it post-harvest because what we're trying to with CCQC to get it registered. In any case, we're doing studies to figure out when it does get registered, where to put it in a pack line. We tried to put it in the pack line over in the beginning during the wash phase with PAA, and it didn't work too well. But those red boxes show that if we put it in after the sodium carbonate tank or the sodium bicarbonate tanks, we can put that on afterward, and we can get both our penicillin decay and our Phytophthora control. The next disease I want to briefly touch on is this sweet orange scab. This is a new disease for California, relatively new. This is a disease that potentially can impact growers. We're currently working with the CRB, the CCQC in the state, the CDFA. Some regulated areas have been already identified as Victoria has already explained earlier that they work on a number of disease detections. And so this disease is potentially really a problem with shipping fresh market fruit because it causes these severe scabbing on the fruit. The life cycle is not really well known. This is a composite life cycle between the common scab and the sweet orange scab. But one of the key things here is it can really cause this formation of the fruit on sweet orange scab. But some of the common scab, which is Fawcettii, can also occur on leaves and twigs. And so this is a composite view of that. So one of the big differences between when we see this sweet orange scab in South America, under high rainfall humidity, you get these severe scabs. In California, the symptoms are more or less associated with wind scarring. And so you can see on the right-hand side, the complete, to me, it's very different type symptoms for what's being found here in California. The method of identification in California is a molecular method. Unfortunately, this pathogen is very difficult to culture. It's very slow growing, and so the state elected to use molecular approaches. And again, they have a common band showing up across all the samples, but then there's 4 or 5 there that actually have a band which are positive for SOS or sweet orange scab. And up to date, there's been 18 different locations identified in California. And these are regulated areas, and they're or the USDA, APHIS wants us to move forward with setting up quarantines in some way. And so we're discussing this at the moment with the CDFA and the industry. So the objectives for SOS, this is a brand new project that we're going to be working on, is to look at the details of culturing this pathogen, molecular identification. Most of you know that molecular detection is not really based on a living organ, it's based on DNA. So we're trying to culture that to show the viability, and then doing the pathogenicity to show the various varieties that are susceptible. We're also looking at fungicides and how to get out of the quarantine. If you have a clean orchard for several years, we can put fungicides on, we can protect the fruit, and we're looking at that as a potential discussion with APHIS as well. The immediate thing that we're looking at is how to reduce the regulated areas to smaller areas. Currently there are a 5-mile radius, and if you do a 5-mile radius for square mileage, it comes out to 78 square miles just with a single orchard around that. So the spores of this fungus are very short-lived. They're very sensitive to sunlight, and so we have evidence already that there's a way to reduce those size of those quarantine or regulated areas. We're also looking at tarping, because bins are already being tarped rather than removing leaves and stems from harvest bins, which is very, again, labor intensive. So if we have that as a condition to tarping the fruit, then we don't have to take out the leaves and fruit from the areas that are potentially identified as having SOS. Okay, the last thing I want to talk about, I only have a few minutes left here, but we continue to work on post-harvest decays. We have some exciting news that we're very fortunate to have move forward during the COVID epidemic here. We didn't have a chance to talk about it, but we are looking at ways to use pre-harvest fungicides for post-harvest disease control, and we are developing new fungicides for post-harvest use. And that's a milestone. This EXP 2020 is a new fungicide, and it's currently going through the IR4 residue, GLP residue program. And so we're already in year 2, and so we're hoping to get this one through in the next 2 or 3 years, and then establish concurrently MRLs, and we'll hopefully have it in a few years as another new post-harvest fungicide. So some of the things we're doing real quick, we're mixing fungicides with these residual sanitizers. We have this sanitizer called EXP-FP, and that's an experimental product. And mixing it with a fungicide, we can control both sour rot and penicillium by reducing the populations in the field. So that's an exciting development. We have a number of different products available, and we have reduced risk materials. Those are in orange. We have conventional materials, and then we have this EXP 2020. It's a conventional material, and we think it'll fit in good in a rotation program. We also have these biofungicides like natamycin and potassium phosphate. The natamycin we continue to work with. It's moving through the registration process with Codex and with MRLs. The reason why we're so high on natamycin is that it's been used in the food industry for over 30 years. And there's never been resistance detected to natamycin in any filamentous fungus. So that's an exciting characteristic. So what we want to do is, you see there, we're mixing it with different fungicides and getting a very good control. And so when it does get registered with our MRLs, with Codex and FATs, we can use this as an anti-resistance treatment. Again, back to the resistance issues. Not only do we have resistance with phosphite, but we detected propiconazole or mentor resistance in some packing houses that were storing lemons long term, and we verified it this past year. And so what we're looking at is that can higher rates of propiconazole work? And the answer is yes. There's other fungicides like EXP 2020 control the resistant issues, and on the right graph you can see that from 150 to 300 ppm we can control both resistant, the orange, and the blue, the sensitive bars. We can control both populations with these new fungicides, so there's no cross resistance. So mixtures look really promising, and we think that EXP 2020 will be a good fit for managing both sour rot and penicillium decays. So I mentioned that mentor works at higher rates, so we put in a 24C, which is a special local need. The current label allows for 2 applications of mentor, which is the label for propiconazole at 32 to 64 fluid ounces. The first is when you go into storage in a lemon packing house, and the second is after storage. So the new special local need that we submitted to the state for green mold and sour rot, which should control the resistant populations, is to do 1 application at 128 fluid ounces going into storage. And then all this fruit that comes out of storage gets washed, and the residues can be brought down, and we've demonstrated that with our residue studies. So we'll have the current label, and we'll also have this 24C label, section 24C, which will allow for 1 application going into storage, and that should control the resistant populations. The last thing I want to mention real quick is that I've been involved in as a system part of the industry that we're looking at new citrus carton labeling. And we've had seminars on these, online seminars. The US and Japan are the only countries in the world that require some list of the potential fungicides that may be used, or a list of the fungicides that could be used on that fruit lot. And again, with trade with Japan, we have to provide that list. There's no requirements for other countries as far as listing pesticides or fungicides being used as a post-harvest treatment. Our goal, as I mentioned earlier, was to deliver really high-quality fruit, and we have these reduced-risk materials and biofungicides eventually available that we can use, and because they're used at low rates, they're highly specific, and there's no mammalian toxicity. This is an excellent opportunity for us to integrate safer products into our post-harvest handling, and then get improved disease control in an integrated fashion. Again, this slide, we had to pick 6. We picked 3 reduced risk materials, the fludeoxonil, azoxystrobin, and pyrimethanil, and we picked 3 conventional materials, thiabendazole, imazole, and propiconazole. This was our pick 6, not to mix with the football goals, but we have a pick 6 of what citrus fungicide we want to list on the box. This is a major issue because in Japan, they have to list what chemicals were put on that fruit at the time of marketing. So we've had SOPP and TBZ and imazole, but we never were able to use all the new fungicides. So now we got rid of SOPP, we kept TBZ and imazole, we added the 4 other newer fungicides, fludeoxonil, azoxystrobin, pyrimethanol, propiconazole, and now this is going to give the industry a lot more flexibility in what tools that they can use. This machine's dying here, so next slide, please. Here we go. Why do we need so many fungicides? I'll just quickly say that every fungicide has different spectrum of activity, has different efficacy, and different aspects like sporulation control. We have this plus-minus chart. So every one of these fungicides has a different characteristic, but we can now combine and use these materials effectively. So this is an example of mixtures. We have imazole, pyrimethanol, or filibuster. These are pre-mixtures. But now we can do tank mixtures with generic materials. We can try fludeoxonil with azoxystrobin and add propiconazole or add TBZ to it. And so again, the service companies, I think for the most part, are excited about this because it's going to allow for greater flexibility and it's going to provide resistance management. Just to demonstrate this, if you have a resistance frequency of 10 to the 4th or 10 to the -7th, that means like 1 out of 10,000 or 1 out of 10 million our spores are resistant to that chemical. But when you mix these materials, you can see the resistance frequency goes down for selecting for resistance to both chemistries. So the benefits of making the change, we're going to improve quality, we're going to have greater ability to have higher quality fruit. We're going to prevent losses and claims. Claims are when you ship the fruit, you've invested in that crop, and then the market pays 90% on what you delivered instead of 100%. And that's a big issue after you just invested in that. So these changes that we're talking about are going to be very important for resistance management and giving the industry greater flexibility. We're calling the ones that we're using as primary fungicides and for disease control, and the secondary ones have to be added because they're on the box, but we're going to use those at much lower rates, infinitesimally small rates. And if the Japanese asked, we're saying we're using all these post-harvest fungicides to optimize quality, and we're going to achieve a good combination, which should be, some will be detectable, others won't be detectable because we're using at very low levels. But again, the Japanese will seem to be accepting this as the new standard for using post-harvest fungicides. I'll stop there, and I thank you for your attention. We were running late, so I'm kind of right on the timeline. Thank you very much.
So do we have a question or two before, right over here?
Phosphates been used to control root.
Phosphates been used to control root rot. They've been shown to have resistance the Phytophthora in the roots.
Yeah, so we've been detecting, there's multiple species of phosphite. And I didn't show all the species, but all the species that I listed there, whether it's Hibernalis, whether it's Nicotiana or Septoria or our quarantine pathogen, Syringe, they're all shifting to less sensitivity because phosphite has been a standard for putting it in the irrigation and chemicating it for root rot control. But it was, and it still is a good strategy. Not every isolate is resistant. You saw those curves. It's just that we're seeing shifts. And if we continue to use these things, we're going to shift them all to the right, which is where their higher resistance is. So we're not telling you to panic and don't use phosphite. We're telling you to use the phosphite strategically in rotation with other materials. The Aranis and the fluoropicolite or soil can be chemigated through the soil just like the phosphites. So you can develop a rotation program using these new materials with phosphite.
So I guess if there's any other questions, you'll be around the rest of the afternoon.
It's hard to stand between lunch
Just many
and
people at lunch. No worries.
Thank you.
Okay, thank you again, Jim.